Okay.
Okay, so we're now recording. Thank you very much, Kerstin, for agreeing to give a Build a Cell seminar, and you can get started.
Great. Thank you very much, Kate, first of all, for having me. And since you asked me to introduce myself, I actually thought I should start at the very beginning. So apparently my fascination for origami started very early on. And if we now fast forward about 20 years, I found Lactate actually during my PhD at the University of Cambridge in the group of Ulrich Kaiser, where I worked on DNA origami nanopores. And this is also where I found out that actually there is a research field which is addressing the questions that I was always very fascinated by, like what is life and can we actually build it? So this is why I went to Germany for my postdoc to Stuttgart with Joachim Spatz. And now since 2 years now, I've been leading the Max Planck Research Group Biophysical Engineering at the Max Planck Institute for Medical Research here in Heidelberg. And in about 10 days, I will also be a fellow of the Max Planck School Matter to Life. And I should say that the application for the Master to PhD program in the Matter to Life school is open for applications right now. So if you're at that stage, or if you know someone who is, we'd be very, very happy to hear from you. Yeah, and with this, I would like to dive into the research part of this talk. So basically, if we talk about engineering synthetic cells, then typically we start by isolating biomolecules from cells, right? Like proteins, for instance, and then really recombining them with the vision to one day be able to really create a transition from matter to life in the laboratory. And now we all know that this is super hard. And now I'm asking if maybe it is so hard because we are trying to build something that looks as closely as possible as the original. And maybe if we were to focus more on function rather than on form, we would realize that abstraction helps and that we would be faster at arriving at the end goal. So I'm wondering if actually instead of using biomolecules from cells, we can build our own synthetic building blocks and really program their assembly to make synthetic cells which may look a bit different from life as we know it, but have the same basic functionality. Because even if recombining the pieces of the puzzle may be really hard, I think it's still possible that if we are open to use new tools and new materials, we can quite quickly build something which has the same basic functionality, but is built from artificial parts. And now, of course, we have to ask ourselves what kind of tools would be suitable for assembling synthetic cells. And I think here it's really important to say, first of all, that there is no reason at all to be dogmatic about this. So it's really fun to constantly be scanning the horizon for new tools that we could be using for synthetic cell assembly. But in this talk, I would like to show to you two tools that we find useful or found useful in the past to make synthetic cells. And then I will show you two functions or discuss about two functions that we realized very recently. So let's start. Why is microfluidics useful for us? Well, first of all, one should say that microfluidics has been developed totally independently of bottom-up synthetic biology. And there are lots of devices that we can simply repurpose to make synthetic cells. So first and most importantly, there are devices to make cell-sized compartments simply by intercepting a water flow here with an oil flow coming from top and bottom. And in this way, you can create thousands of compartments per second. And you can then inject components into these compartments and grow them in the process of it. You can then move on and use other modules to actually divide the compartments, maybe to mimic a basic cell cycle. And then again, you can move on to select compartments for certain properties, kind of an artificial selection mechanism. And finally, release the compartments or 3D print them in a specific pattern. So with all these tools at hand, I think one can really envision kind of an automated assembly line for synthetic cells. They're simply useful for us to move forward, I think. Now, of course, you may argue, I've been talking about microfluidic droplets all the time, which are water and oil emulsions, and they may not be very good mimics of the water and water systems that we would like to have as synthetic cells to enable interaction with the environment or signal transduction across the membrane. So of course, we'd like to have a lipid bilayer enclosed compartment in a water system, so to say. And for this reason, we actually developed a method to transform microfluidic droplets into giant unilamellar vesicles by, first of all, encapsulating small vesicles inside the water and oil droplets, and then basically triggering the fusion of these small liposomes at the compartment periphery to kind of form a 3D supported lipid bilayer at the water-oil interface, and then by chemically breaking up the emulsion by adding a destabilizing agent, we can release freestanding GUVs into an aqueous phase, so to say. So in this way, we can make giant unilamellar vesicles, and in this publication here shown at the bottom, I have tried to explain in very much detail how you can do this by yourself in a lab. So like I'm doing it here. So I hope it may be a useful tool to make GUVs for some applications, at least in synthetic biology, if you want to make giant unilamellar vesicles also with content. The nice thing is that it doesn't really require equipment, because you can make these water and oil droplets by microfluidics, but you can also make them in a very quite simple shaping process as shown down here. So if this may be useful to you, just get in touch if you like. And in the rest of my talk, we will basically use both types of compartment systems, so water and oil droplets as well as giant unilamellar vesicles. So just to point this out up front. So this is why we believe that microfluidics and related technologies are quite useful to make compartments. Why is DNA nanotechnology a good tool for us? Well, DNA nanotechnology, first of all, is about repurposing DNA for construction based on the complementary base pairing of DNA. And with this, others and also us, we've made shapes that are useful for synthetic biology. So for instance, like in my PhD, I made these membrane spanning DNA pores, but there are other examples as well. And what I would like to point out is that it's not just actually about making complex shapes. In the context of synthetic biology, quite often, a very simple shape, namely a DNA duplex, can already be quite useful. So imagine we can tether a DNA strand to the compartment periphery. Then we have an attachment handle for a complementary piece of DNA, which can carry an arbitrary functional group. So how can we tether this? Basically, we often use cholesterol as a moiety which self-assembles into the hydrophobic part of either the surfactants that stabilize water and oil droplets, or also the lipid membranes which we have for the giant unilamellar vesicles. So it's really a simple self-assembly process with which we can modify the compartment periphery with kind of any component that we like, because of the large toolbox for chemical functionalization that is out there for DNA. So we can attach different components, and most interestingly for us right now in the context of synthetic biology is the attachment of cytoskeleton elements, as I also suggested in the title of my talk. Why are we interested in cytoskeletal elements? Well, I mean, if you think about the functions that we perceive as living, right? So be it migration, motion, be it division, for all of these functions, the cytoskeleton is playing a crucial role. So how can we actually reconstitute a cytoskeleton and actually make it functional inside synthetic cells? So as I've shown you, of course, we can tether actin, a cytoskeletal filament, to a DNA duplex and thereby tether it at the compartment periphery. And use this as a linker basically to attach the actin to the compartment periphery. And now, if we add, for instance, ATP as chemical energy, and then motor proteins in the form of myosin beads, for instance, then we can achieve the symmetry breaking contraction of such an actomyosin skeleton, as you can see here. But what I don't like so much about this is actually that, okay, establishing polarity is one aspect of the function that a cytoskeleton has, but there are many, many others. So, and you quickly realize that adding more function means adding more protein, and unfortunately, adding more protein doesn't scale linearly with the complexity, so it gets very, very hard, very, very, very quickly. And this is why we've been starting to think about whether it may be useful to actually build cytoskeletal elements or mimics of cytoskeletal elements from DNA. So really use DNA nanotechnology to establish cytoskeletons for synthetic biology, which have a more diverse function, and in particular can, for instance, influence the cell shape or establish the cell shape and reconfigure it, or also achieve intracellular transport. And now these are two functions that we've been trying to realize with DNA-based mimics of a cytoskeleton. So first of all, let's look at cell shape. Well, somehow, if we want to influence the shape of a synthetic cell, then first of all, in a reconfigurable way, then first of all, we need a strategy to reversibly attach and detach something from the compartment periphery. And this, we thought, okay, which stimuli do we want to choose for the reversible attachment? And we actually thought that pH might be a very good choice, because if you think about it, pH gradients is a universal form of energy generation and storage in living systems. So we thought, well, if we can switch membrane attachment with pH, then maybe we have a way also to go towards autonomous switching cycles. So for this reason, we made use of the such so-called triple-stranded DNA motif, where we have basically a DNA duplex, where a third strand of DNA is wrapped around and to form a so-called triplex, which is stabilized by the Hooke's interaction. If you add a base, this triplex gets destabilized, and this means that the energy barrier for complementary base pairing with a second DNA, which binds here to the hairpin loop, gets lowered, and we can basically form a duplex between this triple-stranded DNA, or initially triple-stranded DNA, and this second DNA strand over here. And like this, we thought that we can switch attachment and detachment to the compartment periphery, where basically at low pH, the triple-stranded DNA is homogeneously distributed inside the compartment, and as we increase the pH, we get recruitment of the triple-stranded DNA to the compartment periphery. And now we tried this, and actually you can see here that if we have the triple-stranded DNA labeled with a red fluorophore, then it's just homogeneously distributed inside the compartment at low pH, and we get attachment at higher pH values, so to say. And now, eventually, we also wanted to visualize the cholesterol tag DNA, which sits at the compartment periphery all the time. So we added a fluorophore label to it. And what we saw was quite surprising to us in the first instance, because what we realized is actually that the pH turning point is shifting to higher pH values just in the presence of this single one additional fluorophore over here. Now, it is known that you can switch the turning point of this triplex motif by changing the DNA sequence, but it was unclear that the same is happening. And hello? Oh, sorry. if you just add a fluorophore moiety over here. So with this, we basically realized that we have to look into this in more detail, because if we really want to control the switching of the DNA triplex, we need to understand this effect. So actually, we set out and tried many different fluorophore combinations at the single-stranded DNA, at the triple-stranded DNA, and so on. And all you need to take away from this plot is basically that depending on the fluorophore combinations, we can get full attachment at an intermediate pH or no attachment at all. So this means that with the fluorophore alone, we can really dramatically influence the dissociation constants and also the equilibrium state of the system. So for this reason, we were puzzled by this. So we did MD simulations in collaboration with Maxime Igaev. We could actually show that what may be happening here is that the fluorophore label at the cholesterol tag DNA is kind of stabilizing a more compact configuration. Whereas if you have no fluorophore, you see that the single strand is extending every now and then, and therefore becoming more available for complementary base pairing with the hairpin loop of the triple-stranded DNA. We then also did kinetic experiments with caged protons, where we basically looked at the dynamics of the detachment of the triple-stranded DNA. really, to cut a long story short, we can say that what we found is that basically the fluorophore label is increasing the effective energy barrier for dynamic switching between the bound and the unbound state, so to say. So this means you really need to pay attention. The choice of fluorophore can alter or completely inhibit the dynamics of DNA structures. But there's also a positive side to it, because we realized that actually you can use fluorophores to really fine-tune the energy landscapes if you want to build dynamic DNA nanostructures. And we think that these conclusions should apply to other modifications on DNA or RNA in more general. If you make similar observations, then maybe these kind of effects could come into play. Yeah, and now that we have basically understood the system a bit better, we set out to actually finally build something which can deform lipid membranes and thereby establish the shape of the GUV. So we made a DNA origami, basically just a DNA origami tile, which consists of 2 layers of DNA and has this triple-stranded DNA modification at the bottom of the DNA. And then this is how it looks in AFM. If you basically induce or allow for stacking interactions at the edges of the structure, then you can polymerize the single DNA origamis, which are maybe 50 nanometer in diameter, into very large micron scale sheets. And now these micron scale sheets have the capacity to actually deform lipid vesicles. So here we assume that the straight sections here that you see on the vesicle are actually corresponding to polymerized sheets of DNA origami. And we also see that we can tune the attachment of these sheets with pH. So we see that at low pH, the DNA origami is not attached to the GUV, meaning the GUV is spherical. But then as we increase the pH, the GUV really gets deformed because of the attachment of the DNA via the triple strand here. And then if we lower the pH again, we can also detach the origami. You should know that there is some hysteresis happening here. So basically, we need to go to a lower pH to achieve complete detachment than the starting pH value. And now you may say, okay, we can trigger this shape transformations of the GUV also in a reversible manner, but not really in an autonomous manner, because we are still adding an acid or adding a base in order to switch the reconfiguration. So how can we get this switching to happen in an autonomous way? Well, we again got inspired by nature, so to say. So if you think about it, mitochondria, so the energy generation units in eukaryotes represent formerly free-living bacteria. So we thought, can we actually top-down engineer E. coli so that they can harvest light to produce proton gradients? So we basically made E. coli which overexpress the inward proton pump xenorhodopsin and thereby change the pH in their environment upon illumination. So that you can see here that this works. So basically, if you stick a pH electrode into the E. coli solution of the engineered E. coli, then you see that if you turn on the light, the pH is increasing, and if you turn off the light again, the pH gradients dissipate and the pH is decreasing again. You can switch this back and forth upon illumination. Now you can also encapsulate the E. coli inside the compartments by microfluidics and observe this pH change with a pH-sensitive dye. In this case, we chose pyramine, and you can see each time the compartments blink or increase in fluorescence intensity. This is basically when the pH increases. Yeah, and now we can put everything together basically and engineer quite a complex reaction pathway where if we turn on the light, the proton pumps in the E. coli become active, the pH is increasing, the DNA origami binds to the periphery where it can polymerize and thereby make the compartment stiff and also deformed. And you can see this here. So basically, if we turn on the light and we take a GUV and we put the E. coli around it, then you can really deform this lipid vesicle in the presence of the E. coli. You can also quantify this. So basically, if you turn on the light, the attachment of the origami goes up. And this happens in each basically light cycle. So here we turn on the light again, pH goes up, attachment goes up again, so to say. I'd also point out that unfortunately we don't get complete detachment of the DNA origami because of the hysteresis effect that I mentioned earlier on. So basically, the E. coli can only go back to the initial pH value, but they cannot really go below it. So this is kind of a downside of the system, I should say, in its current state at least. Yeah. So we can use DNA origami to kind of shape synthetic cellular compartments. Can we also build kind of an intracellular cytoskeleton with which we can achieve transport? So for this purpose, actually, we build filaments from DNA, resembling cytoskeletal filaments that we know in nature. So this is basically a DNA tile design, which consists out of 5 DNA strands, and that they self-assemble into these long, hollow nanotubes. So this design was first presented by Paul Rothman, and we adapted it basically and encapsulated it again inside microfluidic droplets. So here you can see these DNA filaments, and now we encapsulate them inside the microfluidic droplets, and then they are confined to the inner volume of the droplet, so to say. Now, in the next step, of course, again inspired by nature, we would like to make these DNA filaments responsive to a stimulus. So basically trigger their assembly, like we can trigger the assembly of actin filaments, for instance, if we add ATP. So how can we do this? Well, we took the DNA filaments and modified their edges, so that the single piles, and modified their edges with a split ATP aptamer, so that if you add ATP, only in the presence of ATP, we actually get the assembly of the nanotubes. And you can see this in this video, basically. So here we have, in the first instance, non-assembled filaments, and then as soon as you add the trigger, basically they start to assemble into these filamentous networks. We can also quantify the assembly process by measuring the porosity over time. So the porosity for us is simply a proxy for the degree of polymerization, so to say. And you see that if you add ATP to the actin filaments, the porosity is going up over time, and the same is actually happening for the DNA-based filaments. So this really shows that you can trigger the assembly of DNA filaments with natural triggers inside compartments. Since we use DNA nanotechnology, we are of course also free to use synthetic triggers, like for instance DNA strands, where we can first of all trigger the polymerization of the filaments, and then use DNA strand displacement reactions with an invader strand to trigger the disassembly, and then we can add an anti-invader strand, which is basically displacing the invader again to trigger reassembly of the filaments in the compartment. Similar, again, we can use aptamer-target interaction. So this is really just to show how versatile we are with the use of DNA nanotechnology, so to say. Here, for instance, we use nucleoline for the assembly to polymerize the filaments, and then ATP to disassemble them again in this case. Now, I promised that we'd look at intracellular transport, and of course, what inspires us there is these beautiful animations, and also experimental data on basically the movement of vesicles, for instance, on these filamentous tracks, for instance, microtubuli inside living cells. Now, to implement something like this inside a synthetic cell on DNA origami filaments requires some thought. So we actually came up with a quite complex mechanism, and I should go through it one by one. So first of all, if we have our DNA filaments, we basically extended a DNA strand from these filaments, and we created a complement, a so-called chimera strand, which is half DNA. So the green part is DNA and binds to our black strand, and the other half is actually made out of RNA. So this is a so-called DNA-RNA chimera strand. And now, if we have DNA functionalized gold nanoparticles, they are functionalized with a DNA which is complementary to the RNA part of this overhang over here. And now, if we add RNase H to the system, RNase H is known to cleave only RNA in these DNA-RNA hybrids. So basically, this red part here gets chopped off, meaning that basically the track gets depleted, but the gray strand here remains intact, so it's available for complementary base pairing with the non-depleted overhangs that are in front of the gold nanoparticles, so to say. And in this way, we should get a unidirectional rolling motion of the gold nanoparticle along the filamentous track. And this mechanism was first proposed by Yale et al. And termed the so-called burnt bridge mechanism. So in this publication over here, this was demonstrated to work. A rolling motion was demonstrated to work on a 2D substrate. But let's see if it also works in 3D and inside the confinement and on a DNA filament. So first of all, to observe this process, what we did is we modified the DNA RNA chimera strand with biotin streptavidin. This is simply to have a signpost to track the motion. So, and then we looked at it in temp. So first of all, you can see here that the gold nanoparticle is attached to the DNA filament over here. And then what we can kind of see as well is that where the gold nanoparticle has already been, the filament looks bare. And where the gold nanoparticle hasn't yet been, the filament is kind of a bit more textured, a bit wider, a bit thicker, because the DNA, the biotin streptavidin modifications are still sitting all along it. So this is a first indication that this rolling mechanism may actually be happening. But now, of course, we need to look at it inside the compartments. And we didn't just look at gold nanoparticles, we also wanted to implement this with a natural compartment, and then the SUV, like the transport, to mimic the transport vesicles that roll along microtubuli, so to say. So here we have the DNA filament, and we add RNase H to it, and then the same thing should happen for the SUV that happened before for the gold nanoparticle. So first of all, you can see here in the TEM image that also the SUVs are actually attached to the DNA filaments. And here in the STAT image, you see the same thing. So the SUVs are shown in red, and they nicely distribute along the DNA filament, also in the confinement of a droplet. And now, actually, how can we track the motion in 3D? It's not that straightforward. So again, we thought about an indirect mechanism where basically we modify the track with a fluorophore. And basically this means as the SUV is rolling along this track, this fluorophore should get cleaved by the activity of the RNase H. This means that the filament should basically lose the fluorescence, and that the fluorophores, which are previously bound to the filament, should get homogeneously distributed inside the compartment. So we actually tried this, and what you can see here is that first of all we see the filaments labeled, but then the fluorophore is homogeneously distributed at the inside the compartment at the end. Of course, we did all the controls to really check that this is not due to bleaching and so on. We also did controls without RNase H or without the chimera strand over here. And we also made sure that the filaments really remain intact in the process of this. So basically, we released them from the droplets afterwards, and so that not the filaments got damaged, but really just the fluorophore got cleaved off the filament. And now we can again use the porosity as a proxy to quantify the progression of the transport, and we see that the more SUVs we add, the faster the decrease in porosity is happening, for instance. And we can also use this porosity measurement to estimate the order of magnitude for the transport velocity. And this is really just an order of magnitude estimation, but we get about 100 nanometer per minute, which is in relatively good agreement actually with the literature and with what was obtained on 2D surfaces. Now, I've shown you how we've implemented basically intracellular transport with DNA-based filaments or cytoskeleton mimics. Now, can we actually also implement division with a DNA-based cytoskeleton? In order not to raise your hopes, I should say straight away that this is currently not possible, and that we are actually not close at all to establish a division with a DNA-based cytoskeleton. But I can show you in which direction one could be thinking. So of course we know that in cells divide with the formation of contractile rings. So this requires in the first instance that the filaments bundle, form a ring, which then contracts. So if we would want to have something similar with our DNA-based filaments, basically what we need in the first place is we need to bundle them also. And this we can do by adding a molecular crowder. So here you see how the DNA filaments, which are very thin and small at the beginning, the scale by here is 50 micrometer, are really bundling into very thick structures. And here you see in the TEM image that really hundreds of filaments are coming together, greatly increasing also the persistence length of these structures. And now you could imagine that due to the depletion effect, and also basically if you have a lipid vesicle which is smaller than the persistence length of these DNA filaments, you could achieve the formation of ring-like structures. And I should really say that currently we do not observe any closed rings, and we are nowhere near obtaining a contractile ring or a contractile system. However, of course, if one thinks about it, division is really a crucial element towards assembling synthetic cells, right? Like all the processes like evolution, but also much simpler things would require the division of lipid vesicles. And we know where we haven't achieved it, of course, with DNA-based cytoskeletal elements, but also not with the natural cytoskeletal elements. There's very nice work actually trying to reconstitute such contractile rings inside lipid vesicles, but complete division of lipid vesicles with natural or synthetic components has not yet been achieved. So this is why we thought in order to move ahead, until we are there, we kind of need a shortcut towards lipid vesicle division. And we thought basically, okay, we have to go back to the drawing board and come up with something that works now. So basically we thought, okay, what do we need for division? What are the ingredients? First of all, we thought, okay, we have to define the plane of division. And secondly, we need to increase the surface to volume ratio, because obviously two small compartments have a larger surface compared to their volume, compared to the initial one big compartment. And then we realized that actually all we need is already existing. So because there are lipid mixtures which phase separate into a liquid ordered and the liquid disordered phase, and thereby define two hemispheres of a GUV. But there is also osmosis, whereby if you evaporate water, the salt concentration in a vesicle solution is increasing, which means that an osmotic pressure leads to the water efflux across the vesicle membrane. And if you now assume that there is a line tension at the domain boundary between the two phases, this means that the vesicles should constrict and eventually also divide, because then the line tension is removed completely, right? So we actually tried this. And here, first of all, we see a phase-separated lipid vesicle that we managed to form. And then we see if we expose it to an osmotic pressure, it deforms, it deforms in a way that the interface between the two phases is minimized and eventually completely divides, and the two daughter vesicles start to diffuse apart after the division process is completed. Now, because this is quite a simple mechanism, we can actually build a geometrical model of it, which doesn't require any fitting parameters. It's literally just the geometry, describing the geometry of the system. And the nice thing is that with such a model, we get predictions that we can test in experiments. So the first prediction is that actually the osmolarity ratio that we need for division is square root of 2. The time point of division should be independent of the vesicle size. Asymmetric division should happen faster. Any process which is increasing the osmolarity ratio to a factor of square root of 2 should lead to division. I will just show you the first and last. So osmolarity ratio required for division is square root of 2. So basically what you see here is the prediction from the model. So we get vesicle shapes which depend on the osmolarity ratio, and at square root of 2, 1.41, the membrane area is sufficient to lead to two separated GUVs. And we can now define a division parameter, which is basically quantifying the progression of the constriction formation over here. And we can plot the division parameter. We can plot the osmolarity ratio as a function of the division parameter, and we get this kind of relationship over here. And now we can prepare buffer solutions of different osmolarities, and we find that the theoretical prediction and the experiment actually match quite well. And if we repeat the experiment, we also see this in a quantitative way. Now you also realize that actually we've built an osmolarity sensor here, not just this division system for synthetic biology, so we could actually branch this out and make this useful as well. But for us, of course, we are interested in division and how we can trigger it. So we actually thought that if any process that induces an osmolarity change of square root of 2 should be suitable to trigger division, what kind of interesting processes could we choose? So first of all, we chose a metabolic reaction where basically invertase, the enzyme invertase, is breaking up sugars and basically thereby doubling the osmolarity in each reaction step. So basically what we did is we feed the vesicles with a sugar solution, add the enzyme invertase, and then we obtain complete division into two daughter vesicles, simply because the osmolarity after about 20 minutes is reaching this 1.41 ratio. But we can also use photocleavable compounds where we can divide vesicles locally. So we can basically select one vesicle which should undergo division and divide that one within seconds, really. Now, since we used phase separation in order to trigger division, you may rightfully ask, how can you ever get a second division cycle, right? At the moment we can't, but in order to overcome this limitation, we basically came up with a system where we can selectively fuse vesicles of the respectively other lipid types. So basically we are feeding the vesicles, the single phase vesicles after division with a liposome solution.
I don't know. I don't know. Okay. So asking me.
Hello.
I think someone unmuted accidentally. I muted them.
No problem. So basically we have small vesicles here that can fuse to the big one if we add these DNA-based mimics of snare proteins. So basically we are bringing the two lipid membranes in close proximity, and by thermal fluctuations we can eventually get fusion of the small vesicles. And since as soon as the first one fused, we actually have a domain boundary again and the defect, this triggers the fusion. We believe that this is helping subsequent fusion events, and this is why we actually quite reproducibly get phase-separated lipid vesicles back. And we think that in principle, such a system could be suitable to trigger multiple division cycles, which at the moment we haven't shown yet. And I should also point out that this fusion system was first introduced in this paper here, and it was unclear that it works also for phase-separated vesicles, but it clearly does, and we believe that the domain boundary here is actually helping us in the process. Now, you may still say that the need for phase separation is a constraint. So we also looked at the division of single-phase vesicles, where we can still exploit the increase of surface to volume ratio by osmosis, but in order to get to overcome the energy barrier for division, we cannot rely on phase separation anymore. So we need to rely actually on an increase in spontaneous curvature. And we achieve this by adding C6, which is a molecule which is also used as an anti-Fuma agent, and it is acting as a photosynthetizer. So it basically self-assembles into the compartment periphery. And then if you shine light on it, reactive oxygen species are generated, which induce lipid peroxidation. And now this lipid peroxidation is selectively increasing the outer leaflet of the lipid vesicles, and thereby increasing the spontaneous curvature. And this now leads to GUV division as well upon illumination. So with this, we have division mechanisms for single phase as well as two-phase lipid vesicles. And now you may, of course, ask what's next. And for us, the obvious next thing is, of course, to integrate an information storage system. And here we already achieved the division of DNA-containing vesicles with this CE6 mechanism. But of course, eventually, in order to think about eventually also synthetic cells that can evolve, what we would need is kind of a link between the information and the compartment shape. So coming back to what I started with, of the synthetic cells, because obviously evolution can only act on the phenotype. So basically, in order to get towards selection mechanisms and so on, we would need a link between the information and the phenotype of the synthetic cells. So with this, I'm at the end of my talk. I've shown to you two tools and two functions that we realized recently in synthetic cellular systems, and I've shown you why I believe that by making your own parts, so to say, and thinking of new ways rather than just the biological molecules, we may have a shortcut towards actually making a synthetic cell from scratch. And I don't know when we are there, but maybe you know, and maybe this is an interesting point also to discuss. With this, I should really thank my group, especially Kevin, who did the work on the synthetic cytoskeleton, and Yannick, who was working on the division of synthetic cells, and of course also all our collaborators who were involved in these projects. Yeah, thank you very much.
Thank you so much. That is an excellent talk, and it's really, really promising to see how close we are to the vision. You have several questions in chat. Can you see them?
1 second, yes. Should I start from top? Yeah, so Amelia Burns is asking whether it would be possible to cut out the middleman or the middle E. coli and use E. coli pumping channel directly in the liposome membrane. Yeah, that's a very good question, because actually this is how the whole thing started. So when we started the project, we actually thought that this was what we were going to do. So actually we took the E. coli, we isolated the proton pumps, we tried to reconstitute them inside lipid vesicles, and then put the lipid vesicles inside of the compartment so that we can get the proton gradients in this way. And actually this worked, but the proton gradients that we were able to achieve were much smaller than the proton gradients that could be achieved with the initial E. coli bacteria. So this is probably because some of the proteins may be damaged during the extraction process and so on. So I think, again, like in the sense of the shortcut, right? Like I think if we can make our lives, the problem is hard enough, right? Building a synthetic cell is hard enough. So if we can take a shortcut and work with what works, and use this to be quicker at arriving at the end goal, why not? But obviously, isolating the proton pumps would be a very good solution as well. And maybe other people are better at protein isolation and reconstitution than I am. So this may well be a very valid point and a very good suggestion. Yeah, the second question was, what is the yield of assembly and disassembly for the DNA nanotech? What percentage of total liposome population works? So maybe let me go back to the slide. Yeah. So I guess you're referring to the assembly and disassembly of the DNA filaments here. So actually, we quantified this again with the porosity. So we basically looked at the initial porosity, so when the filaments first assembled, and then at the porosity when the filaments are disassembled, and then at the porosity of the reassembly. And actually, one has to say that at least for this cycle, there's no statistically significant difference. So also, if you look at the pictures, you see that the assembly seems to work quite well. However, I assume that, which we currently didn't do, but if you think of ways of going to multiple assembly and disassembly cycles, that eventually you will see some fatigue, especially depending on the systems. Like right now with these two systems, multiple cycles would not even be possible, right? So that's a very fair point, and I hope I didn't misunderstand the question. If not, just unmute yourself if I did. Where was I? Here. When you calculate the parameter for the visit, is there any way to account for the distribution of inside material too, like equal division of the DNA content? So right now, no. So right now, I mean, the fact that we put DNA into the vesicles was the first time we put anything inside whatsoever. So I mean, any kind of equal distribution of the content at the moment, it's short single strands of DNA that are just homogeneously distributed inside the compartment, and therefore, because of the homogeneous distribution, relatively equally distributed in the two daughter compartments as well. But of course, it would be awesome to have like not just a way to calculate the distribution, but also to experimentally really get the separation of DNA to mimic kind of what is happening with chromosomes or something like this. But this is something to dream about at the moment, at least for us. Do people work on lipid synthesis driven division? When you make more lipids and then the membrane grows and then divides. I love the suggestion, and I think that people do, and if somebody is in this audience who does, I would be very, very happy to hear from you. I think it's a really cool way. I mean, right now, as you say, I mean, we are feeding lipids in the form of liposomes, which is almost like boring if you think about it. Of course, it would be much cooler to have the lipid reproduction included inside the lipid vesicle, and then really drive the division from within and make all the parts inside the synthetic cell itself. So I think this is a really cool suggestion, and this is really the direction into which we should be heading. Can we use something to make the actin filaments? Sorry, I think I don't understand the next question.
Which one is it? Oh, can we use TXTL to make the actin filaments?
Sorry, yeah. Or any other shape directing proteins. I guess so. I guess we could be thinking in this direction. I mean, maybe I'm not the right person to ask, to be honest. I think it should be possible, but I have no clue how difficult it is. I would be happy to discuss. So I move on to the next one, unless Harrison, you want to add. So Lauren is asking, do you think larger vesicles are easier to divide than smaller ones because of the membrane curvature or something else? So, I mean, what our model tells us, and what we also verified in experiments, is actually that on the scale of GUVs, the size does not matter. So basically, it's really just the osmolarity ratio in the case of phase-separated GUVs that is determining at what point the GUV will divide. So basically, for the size of GUVs, size does not come into play, but if you go to, for instance, small SUVs, then I think things like curvature really do come into play. And of course, also for single-phase GUVs, it is spontaneous curvature which determines the division. So for the phase-separated vesicles on a GUV scale, no. For everything else, I think size matters, I would say. Could you use something other than DNA for the origami, like different backbone that is nuclease resistant? Yes, I think it would be really cool. I mean, right now we use DNA because it's cheap, right? If we did all the experiments that I showed you with something else than DNA, I think yeah, I would have to ask for much more funding. So in principle, of course, yeah, it would be very interesting to work with like DNA analogs, or also RNA is a very good example, or even protein origami, right? So I think there are exciting directions coming up when we move away from the standard DNA. Is the increase in the energy barrier you were talking about with DNA and conjugate dyes function of the DNA, the dye, or both? In other words, do certain dyes increase the energy barrier in a tunable manner? Let me go to the respective slide. So yeah, here. So basically, we tested different dyes, not just in experiments as shown here, but also in the simulations, and we find that the different dyes definitely cause a differently large increase in this energy barrier. The reason for this is that depending on the type of the dye, it can be more hydrophobic or more hydrophilic. So for instance, Cy3 is comparatively hydrophobic, so it likes to stack in the bases more than say some of the atto dyes. So here definitely you can fine-tune the energy landscape by using different types of fluorophores, so to say. So Cy3 and Cy5, these are really quite extreme examples of how the DNA is compacting and entangling, so to say. What composition of liposomes is best for the osmotic-driven division? So we actually tried I think in the end, 19 different lipid compositions. And we found that if you have complete phase separation, we think that it's quite likely that division can take place. This is what we think at the moment. So basically, if the line tension is so large that you really get vesicles with two hemispheres, we think that the line tension is also then large enough to enable fission of the compartments. Right now, I mean, what we use is always mixtures containing cholesterol and DPPC or cardiolipine, for instance, and the details on that are really described in this Angewandte Chemie paper from last year, 2020. So there you also find an overview of the different compositions that we tested, and if you want to try yourself, then that's really the best place to look. How does the ATP aptamer work to promote DNA polymerization? Yeah, so basically the ATP aptamer is a sequence of DNA which is folding up in a way that it can bind ATP. And now, basically, if you design it in such a way that you have that you split the aptamer in two halves, then in the presence of ATP, the two halves come together, bind, and have basically ATP at their center. And because two tiles come together and bind, this is what is inducing the polymerization. So yeah, basically the ATP is bridging the two aptamers and thereby bridging the two tiles. Were you able to achieve selective transport, or are you considering to try that? What kind of selective transport do you mean? Like with kind of a selective direction, maybe? Or selection of the cargo, so that you're basically transporting one cargo but not the other. We haven't tried it. I think the way to go about this would maybe be to use DNA overhangs of different sequences, so that basically only one of the cargos can bind, and of course only the one that binds could then also be transported. Or for instance, if it binds not to an RNA overhang but to a DNA overhang, then only the one that can be cleaved, so the RNA overhang or so, could bind. So this is the way you could select, but this of course is very simple. So maybe you could think of much more sophisticated and therefore much more interesting things as well. Can you do division on much smaller liposomes, 500 nanometer or less, that are like the ones used in prebiotic experiments? This is a very interesting question. We like to work with big ones for the simple reasons that we can observe them in the confocal microscope much better. So basically, we've been thinking about, okay, what happens if we make SUVs? Do we also have complete phase separation then? Can we also divide them? But currently we actually struggle with proofs, right? How would you prove that this is happening? So Anna, I would be very, very happy to talk, because I think that's a very nice direction in general, to move from the lipid mixtures that we use right now to more prebiotic mixtures, and to look at also smaller vesicles. So I would be super interested in that. And the answer currently is really, I don't know, but it's a really cool and very interesting suggestion.
I think that's all the questions.
I think I
went through all the questions in chat.
Perfect.
I had a couple of questions, but people asked them for me already in chats. I guess my main thing that I'm really looking forward for you to do is coupling TXTL with replication, because then you really are going to be able to go through the whole cell cycle, and it's going to be so cool.
Yeah, that would be very cool. You're right. Yeah.
And I think, you know, when you do those shape changes, and this seems like we're so close to the actual division.
It's a nice suggestion.
And once you do that, I would love to use that system. There's so much evolution stuff we could be doing if we had spontaneous division. So I kind of have a related question. So you said you're looking into division systems with things encapsulated inside. What are you thinking for maintaining the reactions? So let's say you encapsulate protein expression system with DNA and you divide them. You're thinking of ion channels to deliver nutrients, or what's your plan?
Yeah, that's a good question. I mean, I think again, one has to be open to different ways, right? So ion channels are of course a very obvious choice. I should also say that in the division mechanism actually that I presented last, so basically with this CE6 mechanism, when you have lipid peroxidation, you're also making the membrane a bit more permeable. So this could also be ways to make transient pores to get things inside in the end. I mean, the other option would be really fusion, right? So if you think especially about larger cargo, then I think ion channels are of course always tricky, because the larger, because eventually you reach a cutoff, right? So if the components get larger, then and that's a problem. But also, like, really what Anne was saying about the prebiotic lipid mixtures, right? I think some of them were much more permeable to certain cargos. So maybe that's also really a way to go. But with the transcription translation systems, we haven't worked at all yet. So that's really an interesting direction, I think.
Awesome. Okay. If there are no more questions, we're up to the hours. So I want to say thank you again, and thanks everyone for the questions, and see you all next week. Bye.
All right. Thank you.